Okay, good afternoon, everyone. Welcome to another presentation in the TRANSCEND Seminar Series. And again, thank you for joining us. We're fortunate to have Dr. Van Doze with us today presenting α1A Andrenergic receptor activation as a therapeutic strategy for neurocognitive diseases. Basic science research to phase 1 clinical trials. Dr. Van Doze earned his PhD in molecular and cellular physiology from Stanford University in Stanford, California, and is currently an associate professor in the Department of Biomedical Sciences at the School of Medicine and Health Sciences at the University of North Dakota. His research focuses on understanding the role of adrenergic system in the brain and nervous system and neurological disease and disorders such as Alzheimer's disease, epilepsy, Parkinson's disease, stroke, and traumatic brain injury. His laboratory investigates if the modulation of this system improves learning and memory, regeneration, synaptic plasticity, and is neuroprotective. In collaboration with researchers at other institutions, Dr. Doze is helping to lay the groundwork for development of adrenergic-based therapeutic strategies that are currently being evaluated as potential treatment for Alzheimer's disease. Beyond his research, Dr. Doze has demonstrated a long-standing commitment to developing the next generation of scientists through leadership of nationally funded undergraduate research and summer training programs, while also advancing research infrastructure at UND through the establishment of behavioral and microscopy core facilities. Please join me in welcoming Dr. Van Doze.
All right. Thanks for that nice introduction. Okay, most of these talks, when you see these, they are a lot of research focuses on disease and trying to understand the mechanism of the disease, whether it's Alzheimer's disease, Parkinson's, epilepsy. I actually took a completely different approach to this in terms of, I had an interest long ago in see if they could come up with therapeutic agents and a little bit more about translational ideals than studying disease. So just in case we run out of time, some acknowledgments. So I've worked with a lot of individuals through the years, and why I kind of have an interest is that a number of the individuals I worked with actually were clinicians who were involved in either clinical trials, doing clinical trials work, or trying to develop new agents themselves. So it's kind of where I got this, and as you'll see in a minute. So this work started at Stanford University and has continued onward. And I guess this doesn't work. And so the support was first at Stanford University and then continued here in many different ways to support now. I've had a number of graduate students and undergraduate students who worked on this project, and to give what will be the basis for what led eventually to the development of a first in its class of compounds or drugs for, they're soon going to clinical trials for Alzheimer's disease. So I want to acknowledge the ones who are really involved in what this research talked about. There's my graduate advisor, who should pass away, James Porter here. Jim Porter has had a very, very integral role. He actually introduced me to Diane Perez, who in the end is the one who develops the compound and the Cleveland Clinic drug facility there, manufactured the drugs that the compounds are being used. But a lot of this depends on, and to move something, especially from here to eventually translation involves not just physicians, but those other medical centers that have the facilities or transgenic facilities to make the animals that we needed for these studies, as well as the compounds. Now, clinical drug development is a long process, and I'm going to kind of break this into two. Most drugs fail, easily over 90%, and in the case of the dementia probably over 99%. Now, we haven't had a new drug in 25 years, basically, or just one. So you have your so-called, and the whole goal of this is to move something from the bench or the basic science research to the bedside. And you can kind of see the ideals here and how much. Now, a lot of what I want to focus is on the basic science research, and then you'll see how it was moved to to moving to the bedside at this point. Get an idea of these different stages. So first, I will cover some of the basic science research, the bench part, which will involve in vitro studies of brain slices, in vivo studies of whole animals, then some target. And this is where we get into using various both pharmacological methods to activate receptors as well as transgenic mouse models that have been altered to mimic as if you had a compound that activated those receptors. And then once you have kind of a proof of concept, that you then can convince the funding organizations to fund the development of compounds, you have to go through screening, the lead compound optimization, preclinical testing, and finally you start your clinical trials. And I'll mention that. All right, now why I got interested in as a graduate student. So, whereas I say most labs study a disease, whether it's cancer, heart attack, I was interested in the norepinephrine or neurotransmitter, the central noradrenergic. Now it has these characteristics. It promotes vigilance in animal models. It regulates, and also humans, sleep-wake cycle, increases motivation. We knew it modulated seizure activity, increasing the level of norepinephrine in animal models seemed to be anti-epileptic. Increase in norepinephrine in animals as well as humans also seems to have a role in anxiety and depression that can be selective noradrenergic reuptake inhibitors can be used for treating depression. It enhanced learning memory, and then something we later found here was it also regulates neurogenesis. So the interest was, at the time I had interest primarily in what first initially started out was, I was interested in heart actions, and I moved over to neuroaction was epilepsy, because it is anti-epileptic in animal models, but it enhances learning and memory. That's not what our anti-epileptic, this is in the 90s, those compounds did. Primary, one of the largest side effects of the anti-epileptic drugs is that it interferes with learning and memory. Most of them work through GABA or some other system, and so one of the largest side effects with epilepsy was the interference with learning and memory or reducing learning and memory, and yet this compound or this endogenous neurotransmitter you have has other actions. Now a little bit about the system. Norepinephrine is widely distributed throughout the brain. It rises, this is very bright here. Most of the processes come from the locus ceruleus, which is the major center for producing it, and they ramify throughout this is the cross-section of human brain as well as also the rodent brain here. It's activated by stress, gives rise to sympathetic responses, and one of the major targets is the hippocampus, which is involved in learning and memory as well as in adults epileptics. Now, the system releases primarily in the brain. What is released, 90% will be norepinephrine, and maybe as much as 10% may be epinephrine from another set of cells. But primarily it's norepinephrine that's released. And so I thought it was, as a student, be interesting to look at the effects of adrenaline in this section. So this here, and then focused on the hippocampus because it was this horseshoe-shaped region in the subcortical part of the brain. Again, this is a cross-section of a human brain here. Is a part of the limbic system, so it has a role in emotion, and motivation, mood. It's implicated strongly in epileptogenesis, particularly temporal lobe epilepsy in adults. Oftentimes it arises in the hippocampus, as well as learning and memory. And Alzheimer's disease is one of the first areas of the brain that shows the pathology. It receives a very high density of the terminals that release norepinephrine, and it also happens to be in adults one of the major sites of adult neurogenesis and contains neurostem cells. So now adult neurogenesis. So a little on that. It's the formation of new neurons that basically occurs throughout adulthood. Two places in the brain is primarily, one's a subventricular zone of the lateral ventricles, the other is a subgranular zone. Both contain self-renewing, give rise to more neurons, astrocytes, oligodendrocytes, and possibly even interneurons, the inhibitory cells. So I'm just going to quickly give you a little bit of statistics on three different neurological disorders. One is epilepsies. It's a disorder characterized by recurrent seizures unprovoked by any immediately identifiable cause. During a seizure, your cortical neurons misfire and create abnormal activity. Prevents the brain basically from interpreting and processing the company sensory signals. And the sudden surge of activity usually affects how a person feels for a period of time. Fourth most common neurological disorder worldwide. Between 100 to 200 million people in the world have it. Incidence highest in children and adults. It's now increasing with age as we live longer. See it secondary to neurodegeneration and strokes. 1/3 of the people with epilepsy have uncontrollable seizures because none of our treatments do not work that well. And the idea with epilepsy foundations and epilepsy research is no seizures, no side effects. Because basically we have compounds where we can induce you into a coma, stop your seizures or epilepsy, but being in a coma is not the quality of life you can't. Another area is mental health, anxiety, and depression. Again, norepinephrine may have a role in that. The rates in youth dramatically went up in the last 20 years. It seems to be on the increase. I just noticed here that until 17, not so much in individuals my age or more. We saw more with teenagers, I think. But then with COVID, the anxiety and depression went up with everyone. And so now up to as many as 1 in 7 suffer from it. Anxiety is one of the highest points we've seen study. Lastly, I'm going to cover a little bit Alzheimer's disease. 6th leading cause of death in the U.S. You got progressive dementia. 1 in 3 seniors die with some sort of either Alzheimer's disease or another related dementia. It's more than breast cancer and prostate cancer combined in terms of deaths. Someone develops this every 65 seconds. There's 5.7 million Americans. Will be 14 million by 2050 will have interesting deaths from heart disease have declined over the last so many years, and the cost with this disease is going up. In 2018, several of the big pharmaceutical companies discontinued their research programs on Alzheimer's disease. And I'll get to that in a minute. Some of the characteristics, you have tangles, neuronal loss, plaques, gliosis, changes of plasticity, cognitive impairment. And again, I'm not studying the underlying mechanisms for any of these diseases. What I'm looking at is a neurotransmitter, trying to figure out how it has its actions, and can we somehow figure out what new sites or new possibilities for drugs. So again, big pharma stopped. First ones, I think about 2018, Pfizer pulled this, after spending billions of dollars, they decided to stop their Alzheimer's disease research programs. Was it a mistake or not? We'll talk about that in a minute. Today, and just recently in the last week, as you can see here now, Alzheimer's disease related dementias will cost $818 billion this year. It's more expensive. We're spending more on that than we are in both cancer and heart disease combined. It's only going to get worse as our population ages in this country, and it's not just health crisis, but it's economic crisis too. Why the companies pulled out? Basically, there's a greater than 90% failure rate. Actually, it's more than that. I have had a number of relatives, my family have dementia, Alzheimer's disease. 25 years ago or more, they were treated with a drug called Receptor, which basically is an acetylcholinease inhibitor, keeps the, and the same, I have other relatives recently with Alzheimer's or dementia, passed away, and they've been treated with the same drug. Nothing has changed in 25 years. Very little has changed. There is a couple of drugs out now, but for subgroups of Alzheimer's patients. So essentially, so what happened here over this period of 20 years? Over 100 clinical trials failed. Various compounds were, so after spending billions and billions, the companies finally started to switch their funding to other things, other diseases they thought might have a. Now, question is are we starting the treatments too late? Is it because you already have too much death treatment? So, all right. So who stepped in then? Or who kind of stepped in, which kind of, you'll see later on, was actually advantageous for us. There's a couple of Alzheimer's foundations that support research. One of them is Alzheimer's Discovery Foundation. There's also one called Bright Focus. And this is just from 2025. Look at the number of compounds that are in some sort of clinical trial. Phase 1, which is kind of like the toxicology testing, phase 2 and 3. Whole lot of different compounds now. A lot of these, though, now are being funded not by big pharma, but this foundation here is better in smaller amounts trying these long shots. I mean, different things, including ideals like we came up with. So this pipeline is quite diverse. It's still out there. A lot of it is funded. And what will happen then is something that looks promising, then one of the big pharma will often come in and license and run with it. Because to move it further beyond phase 1, where the human toxicology costs, the costs go up dramatically. All right, so a little bit about these new neurotransmitters, or norepinephrine. It works through adrenergic receptors. This is kind of historically, we thought it was just, there was just 3 of them. Actually, historically, they classified them as based on pharmacological reasons, alpha and beta, and later on they separated out α1 from α2 to beta. And some of the things they do is, this is important here because this is a side effect that stops some of the development of the α1 agonists for neurological. And there's α2 and beta. And actually, eventually, though, unfortunately with me and why the collaborations is these individuals cloned and settled on, there's 9 adrenergic receptors. 3 α1s, 3 α2s, and 3 betas. And some of these individuals, Brian Kabilka was at Stanford with me on my PhD committee. He was actually, at the time I was a grad student, they were still figuring out how many they were, and they were then starting to clone them in Diane Perez a little later with the α1s. All right, so now we know, and it's finally settled that there's 9 adrenergic receptors, so when norepinephrine is released, it can act, and along the same line, this cogeneral epinephrine is released, it can activate any of 9 receptors. Seems like a lot of receptors, not unusual for neurotransmitter. They oftentimes have overlapping signal transduction mechanisms. This diversity is observed in physiological responses, and they even have different concentrations of pin. They can be activated at different. So you have tissues or cells will express not necessarily all 9 of them, different ones mediating different actions. They may be activated at different concentrations depending on a role. So most of this talk, we're going to focus on the α1A. Whoops. All right, so we're going to start with what my plan was, started many years ago, was define adrenergic system-based targeted strategies for basically the pharmacologic treatment of neurologic disorders. Now, originally, and the funding came in for epilepsy. And so why this is the cross-section of the hippocampal slice, using a brain slice in vitro model. We knew this area of the brain, and there were various functions out there tied into learning and memory, something known as synaptic plasticity. They thought under critical in epilepsy, one of the areas in adult epilepsy, these seizures will start in this area of the brain. Alzheimer's disease shows some of the early pathology, and it's also one of the 2 areas of the brain that has neurogenesis going on in adulthood. Now, there are a couple major cell types in the hippocampus. Actually, there's many different cell types, but of interest were the inhibitory GABAergic interneurons, which is your major system for underlying synaptic inhibition, controlling the activity of your excitatory pyramidal cells, shown here on the right. And through models, we can actually induce epileptic form. We can burst activity in the brain slice, like you would see in vivo. And so we first started, we knew norepinephrine in vivo was anti-epileptic in the animal models. So at the increase of, and yet in the brain slice, we'd get it some forms of disinhibition. So it was a puzzle. What is going on there? So we had funding from the NIH to look at this. And what we actually found was that norepinephrine was exciting these cells dramatically. It would depolarize them, and it excited them so much that they'd depolarize so much they'd go into a state of what we call depolarization block. They could not fire any more action potential. So it actually was not causing a disinhibition. That was kind of an artifact of the prep to some extent, or there is that, because normally you would never drive them that hard. These cells would crash out because they were depolarized so far, so excited that they could no longer release GABA. To record from these kind of involved the developing new techniques and everything that. Now this is in 1996, we published, Dwight Burgles with the grad student and I, that we looked at what we're looking at here is the firing rate of these cells over here on the left. These interneurons, you put on 6,4-norepinephrine you put a fluorine group on it and it loses its beta properties, so it just activates the α1 and α2 receptors. You can see here there's a large increase in the action potential frequency of these cells, like over here. Down below you can see we're looking at action potentials. And we didn't run experiments in the top here with atipamezole, which was an α2 non-selective blocker. You see the response still occurs, and then the bottom use the selective α1. Now these are not, they're selective for that particular subtype. The α1s are α2, but not within the subtypes of the subtypes, you might say. So it's not, so it just showed that this was an α1 receptor. And we further showed that it was causing the release of a large amount of GABA onto the excitatory cells. This is to the right here. As these action potential, the interneurons would basically increase their activity and release more GABA. And that was a hypothesized that was a way of an anti-epileptic mechanism. So we hypothesized in the mid-90s that norepinephrine's effect, one of its effects for its anti-epileptic activity was activating the inhibitory GABAergic interneurons through an α1 receptor. We didn't know A, B, and C at the time. They were just still cloning them, and the compounds didn't exist to separate it out, neither the agonists or antagonists. So that's where that work stopped until we came here. In the 2000s. Now separately, there was several groups looking at α1 receptors for cardiac reasons. Cardiologists, people in the molecular college departments, and they were developing transgenic mice models to get an ideal. Another way there were not selective compounds, and no pharmaceutical company is going to develop a selective compound, spend that kind of money unless there's a reason. So you need some sort of proof of concept or some reason why you would want to develop an agent, whether you're interfering with the enzyme, activating a receptor, and so there's different kinds of these models were developed. Diane Perez, who had cloned the α1As and Bs, and then there's a D, came up with the idea that she created two different mice models, actually several mice models, where she actually increased the number. She basically changed the genetics of the animal, using different promoters where they would overexpress the α1 receptors. One animal model would overexpress the α1B receptor, and this nickname came from the students working in the lab, and you'll see in a minute. And the other one, it overexpressed the α1A. And this will give you an idea. So what you have here is you have your wild type or normal animal would have, say, one copy, and then these animals would have 3 copies. And the other thing they were made so that when they were activated, they'd stay active longer. So they're called constitutive active mutants. So she made for A and B and even some combinations. And she did it at a level, some labs overexpress like 100-fold. Usually when you perturb the genome, bad things happen. However we got to where we are today, evolution, we're running, actually for any species, running pretty well. So when you would perturb the genome, you'd get bad things will happen. But she set out and she wanted to see, okay, say you had an agonist, say you developed a compound, and she was actually working in molecular cardiology at the time, and this is all for looking at, there's a heart action. It seems to be cardioprotective activating this receptor. Too, it's another story. And they were developing these, and she just overexpressed them the amount as if you had a drug that could activate that receptor on it, what it would do. Other labs had done it 100 to 300-fold, and they got very bad things, and she did too in some way. And that was Diane Press. Paul Simpson out of UCSF, an MD cardiologist, he did a different approach. He was knocking them out. So you could make a transgenic mouse that did not make α1A or α1B receptors or any of these androgen receptors. Oftentimes those are lethal. These were not lethal when you knock out a receptor or something. And then separately, Dwight Burgos later on made one that was inducible, so the animal was normal. When these earlier ones, the overexpression or the knockouts were, I want to say that's the hammer approach. They start out that way as an embryo, so you could be fooling with development and other issues that could change the whole system. He uses a little more clever where it's an inducible, so the animal actually is normal until you treat it with a compound, and then it will stop making the receptor. So you don't so you can wait till adulthood. You haven't done anything developmentally to change the animal, and you can on-off. It'll quit making the receptor, and then you can take that compound away, and then it'll start making it again. Kind of. So this is how one gets into a field or meets other individuals. So Diane Perez had made, she's at the Cleveland Clinic Foundation, overexpressed the α1B receptor threefold, and she got an animal that had neurodegeneration, multiple system, kind of like Parkinson's disease, movement disorder. And like I said, when you perturb the genome, bad things happen. It may even be lethal. The animal, and it had a little shorter lifespan. And also had epilepsy. But there were whole animals, but they didn't actually have anybody who was studying epilepsy. And she'd heard from Dr. Jim Porter was here at UND, just down the hallway from me. He did his postdoctoral fellowship with her, looking at alpha-1 responses and heart, okay, cardiovascular tissue. We got in touch because we were proposing that you activated alpha-1 receptors just to be anti-epileptic. She develops this mouse as if you had a drug, and she's got this phenotype that's like a Parkinson-like syndrome, and this is epilepsy for us to look at it. So she sent us the animals, we looked at them, I looked at the tissue, and they were terrible. The brain slices were terrible. Death and destruction. The tissue was hyperexcitable. It went right along with. So, which you might say, that's blowing up our hypothesis. We're saying that in that area of the brain, it should be anti-epileptic. She's developed this mouse. It's pro-epileptic. It's having seizures. I looked at the brain, and it really is having, she's correct. All right. But that's because we had different, and again, this is all happening at the same time. She develops a second animal where she overexpressed the α1A receptor. Completely different phenotype. No seizures, no obvious disease phenotype of anything going on. And this is below some of the way she built those animals, the genome. So here's the one having a seizure. On the right is the one where she's overexpressed threefold the α1B receptor, and the one on the left she's overexpressed the α1A threefold, and that animal looks like that most of the time. Actually, looks very, very good. No obvious. And this one, So it turns out actually what was the response we found, which we knew was an α1 receptor. And I'll show you the data in a minute, was actually that turned out what we had found was an α1A response. She had been looking at it, originally made an animal for the B. We were looking at an A, and then when she made the A, it matched basically our in vitro results. We're confirming her in vivo results with the overexpression. So these animals were made, and there's ways you can produce seizures in animals. There's different models where you can give them a drug or a gas that produces seizures. One of them is to give them chlorophyll, induces, and you look at the time to the first myoclonic jerk, and then the tonic-clonic. There's some video of it. So when we take those animals, the α1B overexpressed mouse already is kind of prone to seizures. You can see it doesn't take very much. This is the time for it to happen. So you don't have to give it very much of a proconvulsive agent like a gas, all right, to the first jerk and then to a full-blown tonic-clonic seizure. The α1A overexpressed animal, and this is wild type or normal, you have to give it a lot more compound. It had an anti-epileptic phenotype. The animal that had increased alpha-1 α1A receptors. Flip side, I don't show the data, is knockout. When you knocked out the A receptor, those animals, by touching them, you could cause them to have a seizure. So these are animals that no longer have the A. But when you made, and these were made by Simpson, and then Diane copied it, working with him, made them. And when you knocked out the B, remember A is good, B is bad, it also had an anti-epileptic phenotype. Because now you didn't have, there's some sort of yin-yang going on there between the A and the B. So it's kind of interesting, so how people got involved in what you could do. And likewise, in vitro, they were able to show some of these studies initially started in rat, and then a lot of the transgenic work shifted to mouse. That's how they were made. Even you can make transgenic rats, so we switched. So this is going back to rat, and Dr. Porter here and Kristen Hillman, a grad student in his lab, in the in vitro models for epilepsy basically showed this 5-MU as an α1A selective blocker, that it was an α1A in our in vitro model that was mediating the anti-epileptic action, and then she showed that when you block, this is picrotoxin, when you block it, that blocks GABA A responses, that it went away as anti-epileptic action because no longer was the GABA able to mediate its anti-epileptic activity. So even though the interneurons were firing and releasing GABA, it couldn't mediate its effects, showing that. So essentially, she was right, and we were right. What it boiled down to was α1A versus α1B. All right. And then, along those lines, we wanted to see, there was a compound developed some time ago called cerazone, which was an α1A selective agent, somewhat, 3 to 10-fold selective, which for an agonist or activating receptor is not very good. You want 100-fold or more. An antagonist is usually much easier to develop. So. But we did run it through paradigms, and this is more like, it wasn't significant later, but you could see that it was kind of producing, just as animals treated in vitro, and then we're making them with a different agent have seizures. You can see it's kind of producing a little bit of an anti-epileptic phenotype. So that's where that work was, and that will be continued. The NIH has programs where you can actually send your compound to, and they run it through our various epilepsy models for you to see if it will work. All right. Now switching to another neurocognitive disorder, basically your mood disorders like anxiety. Again, we have these animals. So we have this animal, this α1A, we'll call it souped up, and we have the α1B souped up animal in terms of the number of receptors, and we have knockouts. We have knockouts for A and B in our possession now. We have the overexpression, but not super. By the way, when they overexpress the A or the B or A 100-fold, they got bad things, bad heart, bad. And it's not what a drug would do. So this mouse actually has some very interesting characteristics. Besides, so completely opposite. 2 α1 receptors, 1 Parkinson's disease. This one normal, very happy mouse. You're going to see what we find with it. So we ran it through some anxiety and depression models, in vivo models. This animal actually has tail suspension test is, would you look at the time immobile, and if it spends less time immobile, then it's like has an antidepressive phenotype. Now that compares with when they run these models through with drugs that they test for depression they use or anxiety. But so the baseline of this animal it's as if, this is it here, that's what, if you took a normal mouse and you put it on a high-dose benzodiazepine or an antidepressive compound. So its baseline is it has like an antidepressive phenotype. And then for comparison, you can see the knockout, you don't see that. And marble bearing is an anxiety measure, how many marbles they can bury in a certain amount of time. It's really, for mice, it's anxiety. Again, it has this anti-anxiety phenotype. What you would see if you take normal mice and you treat them with the compounds that we use for the, and then we look at the elevated zero maze and light. These also are anxiety measures. And again, this animal has an antidepressive, anti-anxiety phenotype. It's baseline, as if it gives the same response as if you took a normal animal and you ran it through paradigms, or even when you created some depression minutes. All right. So we went in to see if we could, using that compound, Cerazole, which crosses the blood-brain barrier, and it somewhat mimicked that. It gave, again, the antidepressant, but it's not a very. It has a side effects, vasoconstriction and other things, but that would hold it up for human So, and that's another avenue of research in that one could go, continue down, and we're looking at where we were going to make use animals you could make them depressed or anxious and run it through there and see if it worked. And again, so then we also underline some of those actions they think neurogenesis may involve. A little bit with depression, not the sole reason. Also, going forward to learn and remember. So then we want to say, okay, so this mouse has an anti-anxiety phenotype, has an anti-depressive phenotype, it has an anti-epileptic phenotype. You can cause it to have seizures, but it's shifted from normal, and we're still not seeing anything negative with it. So we decided we'd look at neurogenesis. Now, how we got to that, when I looked at those brain slices earlier, I told you with the α1B overexpressed, they're ugly. I mean, they looked like a lot of death and destruction compared to the wild type control when I made it. So when she sent this to the CAM1A, their so-called super mice brain slices, I looked at them, those slices look really good. And looking at them, I just kind of, this is not the way you measure it, but I just thought, they look better than normal, better than the wild type. They just seem to have, when you make a brain slice, that's quite a traumatic brain injury type model you're doing here with hypoxia and everything. I thought there might be more cells, and the tissue just looked cleaner and everything. But then that's just, maybe I'm seeing something. So we thought, okay, let's look at neurogenesis. And lo and behold, those animals actually, and BRD is a way to look at it, we saw there were more dividing neurons compared to wild type in those animals. So we didn't start wondering, and we got the joke going that she had built a better brain in this animal. This is with something you can't hardly see here, but here's some red here. There's different ways of marking cells and ways you're tricky. You can see that there's the stem cells and the hippocampus. This is a diagram where they are, and this is actually 3 or 4 of them shown up here. There are more of them in this animal that are dividing. There's more neurogenesis going on in and this here is showing what this here is. These here, that's got the α1A receptor on them. So you can see they're actually on the stem cells. So the stem from here, the red is actually showing dividing cells, and here done by different lab. So 2 different labs, one at Hopkins and one at Cleveland, and then we're looking, and then we actually measured them. There is a way to properly count cells and see. So we looked at the BRD positive. Now that's cell division. It doesn't mean necessarily once they divide, they have to migrate and integrate into the synaptic circuitry. And so we're not looking so much at that. As you can see here in the subventricular zone, there seem to be more of those cells in the subgranular zone. So there are more dividing cells. I'm not, again, and I thought this visual looking at these, there might be more neurons overall. So this is kind of, at least there seems to be more, suggesting there's more neurogenesis, adult neurogenesis going. So then we tried to mimic that with that Cerazole compound, and we were able to do that somewhat here. All right. So neurogenesis is one thing. So then we're going to look at learning and memory. And again, I'm not looking at disease. And in this case here, are these mice, do they have increased learning? We've got increased neurogenesis. They have an anti-epileptic phenotype, anti-anxiety phenotype, anti-depressive phenotype. They have increased neurogenesis, which you would think would possibly could be. So again, we're going to do these same sort of studies. So we take the α1A overexpressed mouse. Now, wild type, you have this Barnes maze here with these holes, and you have these markings on the wall that so the mice can ornate. So then with light and fans, you motivate them, and when they first start, they'll go all the way around till they find the escape hole. where they can get out of the light. And once they learn that, you have to train them for days. So here's like the training. So you see the cam mice actually even within day one, when you're doing several trials, they're learning quicker where the escape is. B is the normal, and then you have the knockout mouse. So within a few days, it's about as fast as they can run there, they can get there. Now, normal mice will in time also get, once they learn where it's at. It was interesting with the knockout mouse. 40% of them never learned. You could train them every day. It was like this, which I didn't see at the movie, 50 First Dates. The mice could not remember things from the day before. Now that's short-term memory. What is really critical for short-term memory? The hippocampus is basically where things move to long-term through there. So this mouse cannot remember. But it's not all of them. There's issues with genetics and everything with penetrance. It's like 4 out of 10 will not remember the next day or even days later when you go to train. And we ran it through a water motor maze, which I'll actually show a picture of the next one, where this really motivates mice to try to find this platform, and you got these markings around the side here. And again, very quickly, the mouse with the extra α1A receptors learns, and in time the wild type will learn too, but they learn faster, and then there's a max, and the max is, why it maxes out is because how fast it is you can get to the platform underneath the water, or how fast you can get to the escape hole. There's a point here. Again, there's this percentage here about they cannot learn. The other thing is a very difficult experiment to pull because these animals have seizures. So it's very stressful. So putting them in the water, a number of them will drop out because once an animal has a seizure in the water, they're going to die. So you have to pull them out of the experiment. And then there's this is learning, and then there's memory tests many days later. You can see again how much better the α1A overexpressed mouse learns versus the wild type, and then you have the knockout isn't able to learn it. All right. So then, using similar things, we looked at compound cerazolene. We could kind of mimic it too, and that we could increase activating the α1A receptors would increase the learning and memory in normal animals. And just to show you how important neurogenesis is, if you knock out neurogenesis, there's a compound you can use to knock it out here. You see how then they cannot learn. It's almost like the knockout mouse. They cannot learn. Now, this is unrelated to, it's not changing the receptors or anything. We're treating these animals with a toxin that knocks out so cells can no longer divide. So you knock out neurogenesis, animals will not learn. It's compound. All right. Another thing we're looking at, underlies learning and memory, which somebody mentioned, the synaptic plasticity. There's a paradigm where in the hippocampus where if you give a certain set of electrical stimuli, you have a basal response, and then you'll see after that paradigm, you'll see it increased. So normal is it's increased. They call that synaptic plasticity. It's not the underlying learning and memory. The transgenic alpha-1A shows a higher basal, higher level once you do that paradigm than the wild type. But it's a little tricky because you're comparing other animals. You don't really have somewhat of an internal control there, kind of. But you can see for a number of animals, you see this paradigm, which you'll see how this plays out with dementia in a minute here. Okay. Now and then you have these incidental findings or serendipity. I have, as I mentioned earlier, I had a lot of students in my lab, and we'd order in a lot of animals, and everybody's eyes are bigger than their stomach, and they wouldn't get to the animals to use them up. So we ended up with large numbers of, couldn't afford to do these experiments today with the animal care, but the animals that didn't get used. And one day I'm sitting in my office, and the animal behavior research center calls me up and says, "What do you want to do with all these old animals? Should we put them down? Use them?" We'll say, "Well, how old are they?" And they said, 2 years. And I'm like, 2 years? I thought mice normally live only about 18. No, they do, but some will live 2 years. I said, don't put them down. I want to see some. Sometimes you just, and what we found was that this animal the one that has the overexpressed A lives longer than normal. And not surprisingly, the one with the overexpressed B that has the Parkinson's syndrome and epilepsy and everything lives shorter. All right? And it picks a number of animals. And lo and behold, when you do this is on a different background strain, when you do the knockout, it's on a different strain, it flips. The A will live a little longer than normal. The B knockout, which is leaving the A there without the B antagonizing, you might say, the B knockout lives longer because it's not got the A not being antagonized. The A knockout lives shorter, has seizures, etc. Now, epilepsy and stuff doesn't shorten your life necessarily that much, but it can. But you see these characteristics. So all these things are starting to, and now you know why the undergrad students in my lab started nicknaming this mouse super mouse, and the other one we referred to as just bad mouse. So where are we at this point? Whether we're using genetically, we've activating the α1A receptor, or pharmacologically with some compound, you're seeing increased neurogenesis, increased synaptic plasticity, improved cognitive functions, enhanced learning memory, improved mood, these antidepressant, and increased lifetime. By the way, when we looked at those animals, they had a lower incidence of cancer. It was one of the reasons they may have been living longer. There's a whole α1A story on the immune system. So normally, lab mice die with lymphoma, 40% or more incidence. They don't have really cardiovascular disease. You have to feed them really terrible diets to give them cardiovascular disease. So they either will die with neurocognitive actions or cancer, and the cancer rate dropped from 50% to 20% in those animals. So they probably have an improved immune system. So now, but we're gonna look at Alzheimer's disease. We're gonna switch. Now we've been looking at trying to come up with some strategy. We have some possibilities. We've located it. We've got this receptor. No compounds. There's that cerazolone, but it has side effects. You wouldn't want to use it in humans. The animals seem to compensate very well for the vasoconstriction, the blood pressure not being so much a problem in humans. It more likely would be. We know that from other compounds used. So we've had a few AD model mice here from another lab, and we treated some with this compound cerazolone. We had some untreated, then we had some wild type. We just kind of looked at them, and what we saw was just trends. And I don't want to show trend data, but we saw a trend in both mood and learning memory that the animals treated with the α1A agonist, the Alzheimer mice treated with that seemed to be more like normal. But again, the ends were not enough, 4 or 5 animals. And you can see these are long experiments, and they were left from some other treated line, and we just started treating them, so they started showing symptoms. And you see, but one thing is they're not living shorter. This is interesting. Alzheimer's disease mice will live, you can see a little short, but the ends are way too small to make any decisions. But nothing's by itself. So at the same time this stuff is collecting that data, they were moving on another front, and that is we need a compound. They need a drug to activate the α1A receptor selectively. So now you search funding. Now we could go the epilepsy route, which still could do, but my collaborator wanted to go. She had ideas for how to develop a better agonist. She had given some ideas to Roche, a big pharmaceutical company, earlier on how to develop an α1A, and they developed an α1A selective agonist that had 100-fold selectivity, but it did not cross the blood-brain barrier. It was made not to cross the blood-brain barrier. It was used to treat urinary incontinence in women. And actually got all the way to clinical trials. It seemed to work, but then they stopped it because they were concerned about the effects on blood pressure. α1A receptors mediate through the IP3 mechanism increase in blood pressure. The risk they thought was too high, even they weren't really seeing that. They didn't want it, so they pulled the funding, and the group that was doing it was actually going later on do a different company to look at that and move into neural. So how they're going to do this, you're going to have to fund this. So how it starts in 2014, Perez, she's the lead PI, and I submit a NIH grant called α1A adjuvant receptors. We're going to start looking. Our first plan was to cross the CAM1A mouse with an Alzheimer's disease mouse and see if they counteract some of those actions. We actually didn't do it. Instead, I actually gave up my half the grant and let her use it to pay her, because quickly went to developing a compound. And she got funding from the Bright Focus Foundation to develop a new compound. And this falls on to another one, to the Cleveland Clinic funding it, to NIH, NIA World Discovery, in which they have a drug discovery program, to this Alzheimer's Drug Discovery Foundation is now given. Now this is running now, at this date, several million, but already millions, if you looked at it, all went into the basic science. So now we're moving towards the that other part of the. So what do we have? We had this compound. This one on the left is the Roche compound that was developed for urinary incontinence. They stopped it because of concern. It's a partial agonist, not a full agonist. Now, a partial agonist doesn't give you the full maximal effect. The endogenous neurotransmitter full agonist, Cerazolin, is a partial agonist, and this other compound, A61603, is also, it's actually much more terrific. Now, I put it up there because Simpson is launching and gets an NIH grant to develop derivatives of that they, because it, and these, only the one in the center crosses the blood-brain barrier, because these other, some of these other sites, these chlorine on these, prevent it from crossing them. They're too polar. He wanted it for, there's a cardioprotective action, it looks like. So again, this starts out in cardiology and cardiovascular, this whole development of these drugs for that, and moved into neural. Diane, with multiple rounds of grant funding from, and why are the foundations funded? Remember I mentioned earlier, Big Pharma had stepped out of it for a while and started dropping it. The Alzheimer's Disease Foundations started raising more money, and we're funding these long shots. I mean, this is not studying the disease, like underlying mechanism or anything. We're throwing out possible pharmacological, some sort of, because nothing's worked. I mean, 100 trials have failed, billions and billions spent. Pfizer's pulled out, other companies have pulled out. And so they're funding, I want to say, more long shot type things. So we're not studying Alzheimer's disease, but they're willing to fund. And she comes up with this lead compound. And she had advised the Roche about the compound up there, how to make it somewhat selective, and it was. But it then wouldn't cross the blood-brain barrier. She needed to, so take off this chlorine group, some other maneuvers. This then compound will cross the blood-brain barrier. Where's the other one wouldn't? But you're going to have, presumably, still that side effect. And you're going to, interesting how got around that side effect. So she develops a series of, like I mentioned earlier, in that you got compound screening and lead optimization. So this is how we move into translational research, moving it to the bench. You got that compound Cerazolin here on the left, which does, then you got the Roche compound, which this here is preventing it from crossing the blood-brain barrier. Compound 3 actually it has, and I won't show you the data, but she runs into a lot of in vitro models looking at its actions. It's everything from binding, it shows selectivity for the α1A receptor over the other alpha and beta receptors, et cetera. So it's very good selectivity. And you're going to see another very interesting find. And it doesn't affect blood pressure, though. In animal models, this compound did not affect blood pressure, unlike Cerazolin and the partial agonists. And the reason it doesn't is because, which is just a fluke. She set out to develop a partial agonist that would cross the blood-brain barrier and be more selective than Cerazolin. She did not develop a partial agonist. She developed the first-in-class compound, not intentionally, that turned out to act more like a benzodiazepine. It is a allosteric modulator. It actually does not activate the receptor. It binds to the receptor and changes the conformation of the receptor so that the endogenous neurotransmitter norepinephrine enhances its activity. So it's actually kind of like, it's not and it had been hypothesized that there were going to be modulators of G protein coupled receptors, but not too many have been found. So this is not, and I did not show you all the data to how she in the lab characterized it and found out this thing on its own does not have activity, but in the presence of norepinephrine, but not epinephrine, which is what you expect with the positive, and this is a positive allosteric modulator. There's negative, there's all kinds of, it has, and lo and behold, it didn't affect blood pressure, which is kind of interesting. I mean, why not? I mean, what you're going to see in a minute. It enhanced that synaptic plasticity, okay, which is what we saw in the CAM-A mice, and as you'll see in a minute, that is really reduced in Alzheimer's disease mice, and all right, and then is I don't quite know. She also ran it through some behavior models and increased learning and memory in various models. Now, some of this work now is no longer being done by her or me. These foundations, when they start to fund it, it has to be subcontracted out. So you come up with you design experiment to third parties to ensure there's no bias. And they specialize in this, whether it's in vivo, in vitro, and clinical, it is third party. So they are running the studies, they are paid, so the foundation funds you the millions it takes, and then they even tell you what companies to use, which models to use, and their job is to produce the data. So there's no, 40 years ago that might not have been true. Not that we could have done some of these studies, but maybe we really want to see it happen or this and that. And some of our data we hadn't published because I kept thinking it wasn't, I just couldn't hardly believe it. So this is now being done by third-party contractors. And it involves everything from companies in the United States to Europe. And 2 different Alzheimer's disease models in animals. And actually making the compound involved everything from a company in China making it to the Cleveland Clinic making it to a company in India making it. So this is part of the move translation. So what this compound did though, is earlier I showed you how that transgenic mouse, well, LTP in normal, in the background animal here, gives you this response. This is normal. In disease, this is the wild type for this particular disease. And this is a different Alzheimer's that the foundation feels represents Alzheimer's, but it's depressed. You still have LTP, but it's depressed compared to normal in Alzheimer's disease mice. Now these mice have, develop Alzheimer's disease faster, and it's genetic. When you treat those diseased mice with this compound, you see that it almost returns back to normal, to synaptic plasticity. And actually not shown here, but over here, is the compound, that one compound we use to this day in patients, the majority of patients, Aricept, returns to right about here. This actually returns it back more. And that can be seen over here. This bar to the far right is Aricept. This bar, the third one in, is this compound. So you see at different points, it is returning synaptic plasticity. Now this is in vitro model. This is going to follow up with in vivo models too. All right. Then they looked at plasma levels. And you have these that you see in these mice that's related to, and it returns it to normal, these markers that fall with Alzheimer's disease. Now, they don't start treating the mice, they show the symptoms. So it's not like they did it at birth, these mice. They wait to about 4 months in, but they have accelerated. So this compound is returning the markers back to normal. It's restoring synaptic plasticity. Some cognitive functions, I say some just because they haven't been ran through all the paradigms, some of the learning and memory, returning it back, not 100% to normal, but much more closer to almost non-disease level. So how is this thing working? And I won't show you all that data because I wasn't sure if I cut all that. It is a positive allosteric modulator. So what it does, it binds, it'd be the blue ligand here, it binds somewhere on the α1A receptor, and then norepinephrine activates it, and you have the signaling process. And you can have negative, you can have silent, there's different kinds, but this is a positive allosteric, and there's a whole lot of, you can look up her publications the last 2 years, how the in vitro you figure this out, and how she, And it had another fluke trait. It's not just positive allosteric modulator, it's biased. When it interacts with the receptor and the norepinephrine, it actually changes the conformation so it favors, these G proteins can favor different pathways, like an IP3 pathway, adenylate cyclase pathway. It pushes it that it favors an adenylate cyclase pathway, a cyclic AMP pathway, and a P, which is what beta receptors do. We know they enhance learning. It actually, and was not really thought so much could do so. It moves it away from activating IP3. So even though this compound crosses the blood-brain barrier, but again, it's going throughout the entire rodent body, it is not activating the IP3 pathway and causing the vasoconstriction and raising blood pressures. So this thing is, and I'm not saying she did not set out to do this. She was going for a partial agonist, but she recognized it, saw this, I wouldn't say it's a fluke, she found a positive bias. So it's the first in the class compound for this particular receptor, and there's not that many out there for you, that has avoided the side effects that hung up these compounds moving more into human studies. So where is it today? It's a novel small bioavailable positive molecule, selected for the α1A receptor with affinity, and that doesn't bind the other two. Complement selective affects the bound, but not the efferent, and that's what, but in the brain, it's primarily norepinephrine mediates action. There's very little epinephrine, so in the brain, peripherally, epinephrine would have more actions, but it's not, and this is what you see with modulators, allosteric. It's not really an agonist. It does not cause activation on its own, whereas the partial agonist would give you some sort of effect. On its own, it doesn't, but its signaling is biased. It potentiates the norepinephrine cyclic AMP response, which we know can enhance the learning.